Hi, I'm Heather James, conference producer at Technology Networks, and I'm here to introduce our next talk, Personalised Cancer Vaccines, Entering a New Era of Cancer Immunotherapy. I'm pleased to welcome Dr. Mark Yarchoan. Mark Yarchoan, MD, is an Associate Professor of Medical Oncology at the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins in Baltimore, Maryland. Mark runs an NCI-funded laboratory focused on developing novel immunotherapies for hepatobiliary cancers. He also leads multiple clinical trials of immunotherapy agents for those hepatobiliary cancers. A warm welcome to you, Mark. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. Without further ado, I will now hand over to Mark to begin his presentation.
Thank you so much. So I'm really excited to give this talk because I think that this is a really exciting era for those of us who have long believed in cancer vaccines. People have been working on cancer vaccines for really for decades without a lot of successes to show. And I think finally cancer vaccines are starting to actually show real positive effects in clinical studies in patients. So I think really we've fallen forward in this space into a new era. Very quickly, these are my disclosures, and I think of particular relevance to this talk. I am a co-founder of a company, Adventurous Pharmaceuticals, that's focused on cancer vaccines. So I want to start at a very high level and just discuss why people believe that a cancer vaccine is possible. And I want to go back to experiments that were done really in the early 1900s when scientists took mice, injected mice with a mouse tumor, and of course that tumor started to grow. But then the investigators cut the cancer out, and now the mouse was tumor-free. And then again injected the mice with the same number of tumor cells that had been injected the first time around, and mysteriously, the tumors no longer grew. And so it was almost as if the mice had been vaccinated against the cancer. So somehow the mice had learned from the first time that they had cancer how to avoid cancer the second time around. And so there was some sort of memory, which of course now we understand to be immunologic memory, that prevented the mice from again developing cancer. And so this is really proof of principle that it is possible in some way to vaccinate against cancer. Of course, we've learned a lot since the early 1900s. So we've learned how it is that an immune system can see cancer. And I want to talk a little bit about how this works. So every single cell in the body is constantly showing its contents to the immune system through a system called the HLA system or MHC system. So what happens is every single cell inside of the body is filled with proteins which carry out the cell's function. And these proteins are constantly broken down into small little pieces, are put on little proteins, and those proteins are put on the cell surface. And so cells show their contents to the immune system. Constantly immune cells, particularly T cells, go around and survey these little pieces of protein that are put on cell surfaces. And if any of these little pieces is something abnormal, something that is non-self, that causes the T cell to attack the cell containing a foreign protein. So this is how, for example, if a person is infected with a virus, this is how the immune system clears the virus, is that it will go and attack any cell that harbors the virus because little pieces of that virus will be presented on the cell surface. And this same system is actually how the immune system constantly eliminates cancer cells from our body. And so every single time that you or I walk into the sunlight, we're bombarded with UV light from the sun, which causes mutations, and those mutations result in abnormal protein. And so really, if it weren't for the system, every single time that you or I step into the sunlight, we would have a cancer. But it turns out that the immune system is really good at eliminating these very early cancers because the immune system can see these abnormal proteins very early on and eliminate the cancer cells. Now, the immune system is very, very adept at recognizing self from non-self. That's really what the immune system is trained to do. And so it shouldn't come as a shock to anybody that the more mutations a cancer cell has, the more foreign that cancer cell is going to appear to the immune system. And so the more likely the immune system is to actually attack that cell. And this has been sort of shown in many different ways, but one of the ways that I like to talk about this is just looking at the landscape of different cancers that exist um, and their response to what I will call the first generation of cancer immunotherapies, which are drugs called checkpoint inhibitors that broadly turn on the immune system. And you can see that these drugs called checkpoint inhibitors have really revolutionized the treatment of many different kinds of cancers like melanomas and other skin cancers, as well as some genetically defined subsets of cancer that are just packed with mutations. These cells are hypermutated, have lots of mutations, have lots of abnormal proteins. And so when you give a drug that broadly takes the brakes off the immune system, the chance of success is very high. Conversely, these checkpoint inhibitors have really not, in general, worked for these cancers with very low numbers of mutations. For example, pancreatic cancer, where the cancer cells really look much more like self, and so they're much less likely to cause effective anti-tumor immunity. And I think that this idea that mutations and the abnormal proteins that mutations code for. I'm really sorry. Can I pause for a sec? And at a very high level, I think this concept that mutations and the abnormal proteins that mutations code for, that's really the driver of anti-tumor immunity is really exemplified by this really pivotal clinical trial that was done that took patients with colorectal cancer. And colorectal cancer is, for the most part, a relatively low number of mutations cancer, but there is this small subset of colorectal cancer called mismatch repair deficient colorectal cancer that has lots and lots of mutations. And this is, for the most part, colorectal cancer that come as a familial syndrome for patients that have something called Lynch syndrome. And actually, this same phenotype of hypermutated cancers can be found in other tumor types beyond colorectal cancer. And in this case, the investigators took a group of patients who had colorectal cancer. Some of them had this genetic makeup of a very high mutation burden cancer, and the majority of patients didn't. And they gave them all anti-PD-1 therapy, which is, again, these drugs that broadly turn on the immune system. And what you can see is that, in general, the patients who had this marker, this mismatch repair deficient marker, most of their tumors actually melted away, and many of these patients actually were cured from widely metastatic cancer. Whereas the colorectal cancer patients who didn't have this marker tended not to have responses, as you can see. So each of these bars on the right here, each of these is a different patient. If the bar is up, that means the cancer grew. If the bar is down, that meant that the cancer shrunk. And you can see really the patients with the mismatch repair deficiency in black or blue were the ones who really benefited from the checkpoint inhibitor. And the patients who didn't have this marker did not tend to benefit. And actually, based on this study, in the United States, the FDA approved a checkpoint inhibitor called pembrolizumab, marketed as Keytruda, for anybody with any cancer that had this mismatch repair deficient marker. And so this was really a pivotal moment, I think, in the field, because it was the first time ever that a cancer drug was approved for any cancer based on a biomarker. So up to this time, drugs were approved for example, lung cancer, or were approved for kidney cancer. In this case, the FDA actually approved pembrolizumab for any cancer with this marker. And so it's called a tumor agnostic biomarker and a tumor agnostic approval. And I think it heralds the way that the field is moving, that we really are starting to see cancer as a genetic disease. And this is one of the patients who participated in this early study. Her name is Stephanie Joho, and she was really in very rough shape. She had had multiple lines of treatment for her cancer and really was told that there was nothing left to offer, but then came and participated in the study and ended up having a complete response and is now hopefully cured of this cancer and has really become a spokesperson for cancer research and immuno-oncology. So I want to fast forward to cancer vaccines. So cancer vaccines are a newer form of cancer therapy. As I mentioned at the very beginning, people have been trying to develop cancer vaccines for decades and really haven't had a lot of success. And I think this is largely due to the fact that until very recently, cancer vaccines were developed against cancer-associated antigens. But we know, as I've shown you, that most of the reason that the immune system attacks cancer is that cancer contains mutations. Mutations code for abnormal proteins, and those abnormal proteins are seen by the immune system. And what that really tells us is that if we're going to stimulate anti-tumor immunity with a vaccine, we really want the immune system to see those abnormal proteins. But because those abnormal proteins are different in cancer to cancer to cancer, it really means that cancer vaccines may need to be personalized for an individual patient. And until recently, that hasn't been possible because it's just sort of technologically impossible to figure out what are the mutations in the cancer and make a vaccine for one patient. But technology has really improved. The cost of sequencing have plummeted, and we have new vaccine platforms. And so this study that I'm going to present here, which is really hot off the press, an update was just given just a couple of weeks ago at ASCO in 2024, was really the first randomized study to test the idea that we can make a vaccine for an individual patient targeting those abnormal proteins and make the immune checkpoint inhibitors that have become sort of common denominator of cancer care work better. So in this case, patients with melanoma, which is a form of skin cancer, underwent surgery for melanoma. So these are patients with earlier stages of melanoma. And then these patients were randomized to either receive the standard of care, which is pembrolizumab or Keytruda, which is, again, one of those immune boosting drugs that takes the brakes off the immune system, versus pembrolizumab plus a vaccine. And this vaccine is personalized for an individual patient. So they take the mutations in the cancer and they actually make a vaccine that specifically gives up to 34 of those little pieces of abnormal protein that the immune system would see back to the patient. And this is, again, a relatively small study, but as you can see here, the patients who got the vaccine were less likely to have their melanoma, their skin cancer, come back after surgery as compared to the patients who did not get the vaccine. And so, I think this really provides, I think, fairly compelling evidence in human patients that cancer vaccines can really work in a way that is really quite striking. I mean, we don't tend to see curves separate like this in oncology. So what you can see is looking out to, for example, 30 months out from surgery, 75% of the patients who got the vaccine plus the pembrolizumab did not have their cancer come back, compared to about 55% who got the pembrolizumab without the vaccine. And so really the risk of the cancer coming back was halved by the addition of a vaccine. So this is really a watershed moment for those of us who have believed in cancer vaccines. And I think it shows the importance of targeting those abnormal proteins, those neoantigens that come from mutations in the cancer. Now, if immunotherapy is going to work, melanoma is a great tumor type to try immunotherapy on because it has such a high number of mutations. It's caused by UV light, which causes lots of mutations. But I think the question is, can these vaccine approaches work for cancers with fewer mutations? And again, the jury is not out about this, but there's initial signs that actually cancer vaccines may work beyond melanoma. So this is a trial done by colleagues at Memorial Sloan Kettering that used a vaccine made by Genentech and BioNTech that was personalized for patients who had pancreatic cancer. Now, you'll remember when I showed that slide about number of mutations that pancreatic cancer was at the very low end, so a very low number of those neoantigens, those abnormal proteins. So patients received, in this case, a checkpoint inhibitor, atezolizumab, with a personalized vaccine. This was a single-arm study, so everybody got the checkpoint inhibitor plus the vaccine. But what was intriguing is that the patients who mounted a response to the vaccine, so when the investigators looked at these patients' bloods, the patients who had clear immune responses against the proteins that had been given in the vaccine were much less likely to have their cancer come back after surgery than the patients who did not mount a response against the vaccine. And so again, this is a small study. It's non-randomized. There are other potential explanations for what I'm going to say, but I think the most likely explanation here is that the vaccine worked the way that we really expected it to work, where patients mounted a response against those abnormal proteins and then went on to kill those abnormal tumor cells that contain those same proteins, resulting in pancreatic cancer being less likely to come back after surgery. And then, the obvious question is, will this only work in patients who have very, very little cancer? So the patients who have so-called micrometastatic disease, little specks of cancer floating around after surgery, or can cancer vaccines work in patients who have more tumor, like patients with established cancer? And, again, I think the jury's not totally out about this, but I want to present a study that we were involved in that looked at treating patients with liver cancers and gave patients a drug called pembrolizumab, again, one of those standard checkpoint inhibitors plus a personalized vaccine. And these are patients who actually had active cancer, so patients who had established tumors. And what you can see is that about a third of the patients who got the vaccine plus the checkpoint inhibitor had their tumor shrink in a major way. Those are patients who have the blue lines here. Each of these lines is a patient, and down means that the cancer shrunk. And several of these patients actually had so-called complete responses, where the cancer completely went away and was really undetectable. And so the obvious question is, did a third or so of patients who respond, did they all respond to just the pembrolizumab, or did the vaccine actually contribute to their responses? And again, it's impossible to know for sure because there was no randomization in the study. Everybody got the combination, but the response rate to the pembrolizumab alone is in a range of about 12 to 18%, depending on what study you look at, whereas this response rate of almost a third of patients is clearly higher than that. And so it provides additional evidence that maybe vaccines can really work, not just in the patients who have micrometastatic disease, but also in patients who have more established disease in this case. And this is just an anecdote from the trial, but I think it really confirms a lot of the concepts that we've talked about today. So you can see this is a patient with advanced liver cancer who participated in the study that we just talked about. And this patient underwent a biopsy of the liver lesion that you can see with the red arrow here on the left. And then a personalized vaccine was made just for this patient, just for that tumor. And the patient actually started to have a wonderful response where the tumors in the liver started to shrink. But surprisingly, there was a spot in the adrenal gland outside of the liver, and that's that yellow arrow, that didn't respond at all to the treatment. And so this is, not unheard of for some parts of the cancer to respond and some parts not to respond, but it's a little bit unusual for this to happen. And especially for an immune response, usually you expect if the immune system is going to attack cancer, it's going to attack cancer everywhere. And so we tried to understand, how did this happen? And so what we did was we actually biopsied the adrenal gland, the one that did not respond, and looked at the genetics of the tumor that didn't respond versus the one that did. And what you can see is that even though this was clearly the same cancer and many of the mutations were shared, so about 16 of the mutations were shared, the adrenal gland had, this is the one that didn't respond to the vaccine plus pembrolizumab, had some mutations that were not present in the liver lesion. And conversely, the liver lesion had some mutations that the adrenal gland didn't have. And you'll have to remember that the vaccine was manufactured specifically to target the abnormal proteins in the liver tumor. And what we found was that the only proteins that really generated an immune response for the vaccine were the ones that were only in the liver lesion and were not in the adrenal gland. And so I think what that shows is that as cancers become more advanced, you develop tumor heterogeneity. And this is really going to be a barrier for cancer vaccines and immunotherapy in general, that we can get very strong responses against the mutations in one part of the tumor, but if a different part of the cancer has different mutations and different abnormal proteins, the efficacy may not transfer over to all parts of the tumor. And I think this is an emerging challenge for our field. So with that, I just want to acknowledge a lot of my colleagues here at Johns Hopkins, where I practice, who help me with various aspects of the work that I presented, and organizations that fund our research. And happy to take some questions. Thank you.
Thank you so much, Mark, for such a fascinating talk. And as with all of the talks, we're likely to have lots of questions from the audience. So I can see that we've received some already, and I'll move straight into the Q&A. So the first one is, how did all of the funding and research on COVID vaccines affect the development of cancer vaccines, and what learnings from COVID are relevant to cancer?
Oh, that's such a great question. I glossed over this, but the trial that I presented for the patients with melanoma, the skin cancer that was cut out, that where the vaccine prevented recurrence, and the pancreatic cancer trial that I presented, those trials used vaccines generated from Moderna and BioNTech, which of course are household names now after the COVID pandemic. What I would say is that to generate these personalized vaccines, one part of the equation was to be able to really figure out the genetics of a cancer very quickly and at a reasonable cost. But the other part of the equation is having a vaccine that you can customize for an individual patient. And really, I think RNA and other platforms that were really, I think, advanced as a result of COVID have proven to be really powerful tools in the fight against cancer. And so, obviously the COVID vaccine is not personalized for every single patient, but I think that the COVID vaccine validated that these platforms are feasible and can generate strong immunity. And so now these newer vaccine platforms that were promoted as a result of COVID are now turning their attention to cancer. And so it's very, very exciting. I really don't think we would be where we are without the COVID pandemic. So, there is an upside to a lot of what's happened.
Excellent. Thank you for such a comprehensive answer there. And I've got time for one final question, I believe. So, do you think that these kinds of cancer vaccines can also be used for prevention of cancer?
Yeah, so most of the excitement in the cancer vaccine field right now is for personalized cancer vaccines. to make a personalized vaccine for an individual cancer, you first need to have a cancer to sequence um, and uh, and so the sorts of approaches that I've discussed today, for the most part, may be very powerful for preventing cancer from coming back after surgery, but are unlikely to be the answer for patients that don't yet have cancer, because again, you wouldn't even know what to include in the vaccine. Um, that said, we really do think that cancer vaccines are going to work best the earlier they're given in the cancer life cycle. So the analogy I would give is with COVID, we don't give COVID vaccines to patients who are sick in the ER with COVID. We give them preventatively. And I think the same thing is going to be true of cancer vaccines. The earlier you give them, the better they're going to work. I haven't talked about this today, but I think that some of these abnormal proteins that are used for these cancer vaccines um, are shared from cancer to cancer to cancer. And so there may be a role for cancer vaccines targeting some of these abnormal proteins before patients even develop them in the early initiators of cancer. So more to follow, but I think it's a very exciting area that vaccines may go.
Wonderful. Thank you so much, Mark. Unfortunately, that's all that we have time for today. Any questions that we didn't have time to get to will be sent to Mark after the event. As with all the other talks in this event, this talk will now be available to view whenever you want for the next 30 days. And that just leaves me to say thank you so much again, Mark, and we'll be back soon with the next talk.
Thank you for your time. Thanks for listening.